feel free to put.
All right, so thank you very much for the introduction, Charles. So as he said, today we're going to be covering hybrid yeast and what that actually means for the brewing industry and what some of these hybrids can do for you. So what are some examples of breeding that you may be used to? It's been used for centuries in agriculture and animal breeding. So a really good example of that is dogs. The difference between a Great Mastiff and a Chihuahua. They're both dogs, but they've both been bred for very specific purposes, and they have very starkly different phenotypic characters. Example in the brewing industry where breeding has been used for decades is hops and barley. So plant breeders have been developing new flavors and aromas of hops. We're getting some of those new citrus and tropical style aromas from these breeding programs. And in barley, plant breeders have been breeding new barley to increase yield, pesticide resistance, increasing protein levels, and then obviously manipulating flavors and aromas. But a lot of these breeding programs in agriculture can take 8+ years for commercialization, because if you can think about it, they go with the breeding season or the growing season. So you take a hop or a barley and hop and you grow it throughout the season. You figure out which produce the best result, and then you take it, you carry it forward the next season and breed it. So you can go through 8+ generations. So it takes a very long time. Whereas when we do it with yeast, we can do it within a matter of hours. So we're going to get into some of the practicality of how we actually breed yeast. And it's actually done with a microscope. There's no chemicals or GMO techniques involved. It's literally just what we call the Cadillac of microscopes. And this is an example, a picture of our lead research scientist, Zachary Turgeron, and he's using the specially designed microscope that is affixed with a special holder where you can hold an agar plate, and it has a camera on the top so we can visualize things on a screen. And it's also affixed with a specially designed little joystick where he's able to actually pick up a single yeast cell and move it around on the agar plate. So brewers are very familiar with propagating yeast, and similar to when you grow bacteria, you're just creating a genetic clone of the yeast that you're growing. Whereas what we do is we form yeast spores, which are then able to sexually mate, and we create something that's a 50-50 hybrid of the two parental strains that you start with. Whereas you're looking for consistency in your propagations, when we breed yeast, we're looking for genetic diversity. And we do this by growing the yeast in a special media, because when yeast are stressed out, they'll form the haploid spores, which we're then able to breed. So we grow the yeast in a special media that's very low in nitrogen and has an unfermentable carbon source, and the yeast gets stressed out, forms a haploid spore, and then we're able to breed. This is a zoomed in picture of what the agar plate and microscope actually looks like when we're breeding. So if you look right in the center, you can actually see the hair-like needle, and it allows the scientists to pick up a single spore. They'll move it over and put it directly beside another yeast spore, and if the cells are of opposite mating types, they can actually breed. So again, there's no chemicals or GMO techniques involved. It's literally we pick up yeast and put them beside one another. Like I had said, that special microscope is actually affixed with a very nice camera, and this is actually what it looks like under the scope when we're breeding. So we can start with a spore pair, and to give an example, we could start with, say, an American ale strain that has a really nice kinetics profile and it attenuates very well, but it has a neutral aroma profile. whereas maybe you want to make an IPA or a New England or something that's going to be paired with a really nice hop, and you want to pair it with a yeast that has a really elegant ester profile, or maybe produces some beta lyase enzyme where it can actually accentuate some of the hop aromas. So you pair it with a Sauvignon Blanc wine strain. But as brewers know, Sauvignon Blanc wine strains, or wine strains in general, don't consume maltotriose, so they don't attenuate very well. So that's why you'd want to breed it to the American ale strain that has the high attenuation. If the cells are of opposite mating types, they'll actually start to move towards each other on the agar plate, and that's what we call schmooing, just like schmoozing at a party. They'll start to move towards each other through cell signaling and cell pheromones. You then get cell fusion, where the cells will actually fuse their cell membranes, and they'll start sharing a cytoplasm, so everything inside the yeast starts coming together. You then get migration of the DNA material that's in the nucleus, and you get the DNA material fusing together, and that's with the karogram, and then you get your end hybrid. And hopefully this hybrid is the perfect 50-50 mix of the high attenuation of the American ale strain, and then that beautiful, elegant Saison, or the wine hybrid aroma. But just like when you have a child, you can't necessarily predict what it's going to look like. And if you have multiple siblings, they're not all going to look the same. We end up having to do dozens of these spore pairs. And then we do small scale beer fermentations with all those hybrids, and we actually look for the traits that we want. So we'll make beer fermentations, we'll check the attenuation to make sure it's good, and then we'll also do sensory profiles and make sure it has the sensory that we're looking for. So what are some of the selectable traits that brewers might be interested in that we can actually look for in the lab when we're breeding? Attenuation, so increasing or decreasing. We can change flocculation characteristics, so increasing flocculation or decreasing. We can change the speed that the yeast ferments at, so making it ferment faster or slower. A big one for craft brewers nowadays is flavor and aroma. So looking to make new distinct styles of beer, or just enhancing certain esters and aroma to pair with your hops and barley. Another one OL is stress tolerance. So we can make yeasts that are more thermotolerant or more ethanol tolerant. So producing barley wines and IPAs. We can do also sulfur reduction. So a lot of those sulfur off aroma compounds can produce really nasty off aromas at really low sensory thresholds. So it's really favorable for brewers if we can remove those. We can also change temperature profiles of yeast. So looking at potentially making an ale strain that can ferment at lager temperatures, or vice versa, a lager temperature that can ferment at warmer temperatures, and maybe you can create quicker tank turnarounds. We can reduce VDKs of yeast as well, so potentially producing lower VDK lagers or English ales, again, increasing your tank turnaround time. And we can incorporate enzyme activities into yeast, or we can remove enzyme activity in yeast, which is an example that we're going to go over right now, where we partnered with Lallemand to create this farmhouse hybrid, which is a non-diastatic Saison yeast, which I'm sure most brewers know. Saison yeast produces an enzyme called glucoamylase, which can actually chew away at some of that unfermentable dextrin material in beer, and which causes a lot of issues with overpressurization of your packaged beers. You can get exploding bottles, overpressurized cans. So it's not only ruins your beer, but it's also a safety issue for consumers. So looking at farmhouse, some of the key traits that we're looking to breed: it's a hybrid, of course. It's not diastatic, so we remove those STA1 genes that create the glucoamylase enzyme. We also wanted to make sure that it had the proper Saison flavor and aroma. which Sylvie and we have some brewers that will be joining us to talk about more of the Saison flavor and aroma that the farmhouse creates. And then also we bred in reduction of hydrogen sulfide aroma because we wanted that Saison flavor and aroma to really shine. So we didn't want any of those rotten egg off aromas. And this is how we accomplish that. So these are the parents that we use to breed farmhouse. It has 3 parents. The first, we started with an Italian yeast that was identified in an Italian vineyard by a master's student from UC Davis. And they were doing some bioprospecting in Italy and brought hundreds of yeast back to their lab. And they identified this one yeast that was very special, and it just naturally makes no hydrogen sulfide. But it has, in beer, it has very poor attenuation levels because it's a wine yeast. So we bred it to a yeast that we discovered in Vancouver through our bioprospecting program that has very high attenuation levels and also a very good ability to breed. So we bred those 2 yeasts together. We ended up with a hybrid that had high attenuation levels, no H2S, and no STA1 genes, so it doesn't over attenuate. But it was neutral in its sensory profile and didn't have any of that characteristic kind of spice and floral and fruit that you want from a Saison yeast. So in our sensory program, we went through our culture collection, and our sensory program identified a really nice French Saison that had very high ratings in its sensory characteristics. So we bred that to the hybrid, and we ended up with farmhouse, which now had no diastatic activity, so it's not going to overpressurize in your packaged beers. It had no H2S, so it's not going to have any of that rotten egg sulfury aroma. And it has a really nice, elegant Saison flavor and aroma. This is a brewing trial that we did in our lab, and you can see this is a gravity chart. You can see in the blue line is the traditional STA1 positive Saison, and you can see it has that characteristic Saison diastaticus curve that just never seems to end and just keeps going and going forever. Whereas with the farmhouse, it reaches terminal gravity where you could package the beer on day 6, typical more like an English ale or an American ale. And that's because the farmhouse, again, doesn't produce any of that glucoamylase that is slowly chewing away at the dextrin material and liberating glucose, which then gets turned into CO2. So in addition to just having quicker tank turnaround and being able to produce these beers a little quicker, what does it actually mean for the packaged beer? Because I know that's a big concern for a lot of brewers that are using traditional diastatic strains. So we did a study in our lab, and we looked at what a typical profile of a Saison beer looks like when it gets packaged. And it has roughly 10 grams per liter of additional fermentable sugars or activity that could happen in the actual packaged beer. And that equates to roughly 4.6 grams per liter CO2, which translates into 2.3 volumes. 2.3 volumes may not sound like a lot, but when you put that on top of the 2.5 that's already in the beer, you're talking, close to 5 volumes, which is definitely reaching the pressure limit of a lot of glass bottles. So you're definitely going to have foaming beer and potentially some exploding beer. But because we've removed the STA1 gene, you don't have this worry producing Saison beers with the farmhouse strain. The dreaded worst case scenario, what would happen if your diastaticus or Saison strain, traditional Saison strain, got cross-contaminated into an English strain that had a lot of residual body, or a lot of residual maltotriose? So you brew a Saison beer, you potentially don't perfectly sanitize your tank, and then you brew an English beer. You're looking at potentially 35 grams per liter of fermentable sugar. That translates into 8.1 volumes of CO2 in your packaged beer. And you're certainly going to have some potentially exploding cans and exploding bottles, which is a pretty big consumer risk and probably a pretty big headache for your QC departments. But with this yeast, we've basically eliminated that issue. I'm going to now pass it over to Sylvie, and she's going to talk about some of the field trial results that Lallemand has been getting with the farmhouse strain.
Yeah. Thanks, Jessica. So in this slide, and you can click 3 times, I think this, unless, no, actually, I think the arrows are hidden. Yeah, just go back to the previous slide. So this slide highlights the results of 15 different trials that were carried out around the world. The idea was to collect as much information directly from breweries to be able to, before we launch the product, to be able to advise brewers as much as we can on what the yeast can do. The idea is not for you to go and look at all the data in details. It's really to show you how much, if you look down at the line that is in orange, you have the different flavor outcome and aroma coming from all those different brews, and they all, you know, slightly different, and I think that's expected. If you look at the top line, you have an original gravity that is different for each of those trials. You have temperature that is different from each of those trials. You have the attenuation level that is different as well. So depending on also on the vessel that you use, the volume, etc., you will achieve different results. Bottom line is all of these were extremely well received. And again, you can use really all those parameters to your advantage and create the beer that you desire. Jessica has mentioned that obviously this yeast hybrid, being non-diastaticus, will not attenuate as high as a saison, as a diastaticus saison, but still it was bred to reach high attenuation. Now, if brewers want higher attenuation, there is also ways to achieve a drier beer by either adding enzymes in the mash or adding glucose in the during fermentation, and that will reach a higher attenuation. And some of them have done this. Here, you can see T1 has added enzyme and reach 100% of attenuation. So anything is possible if you know how to do it, and obviously we are there to advise on how to use this strain. Next slide. We're going to focus on a few trials where we thought they were extremely relevant data, and we could draw some conclusion on how best use farmhouse to get specific flavors out of it. So this is the trial 14 from the slide before. So this trial was carried out in Canada. You can see the details of the mash regime, the type of malt that were used. You have some wheat malt in there, and the hops that were used. And the evaluation, the organoleptic evaluation of the beer was done by 13 people. And on the right, you have the average result of those people that tasted the beer. So you can see that the score is very high, of 4, and you have the intensity of the fruity flavor. Now, what's important in this trial is the temperature was in the low 20s centigrade, and at that temperature, the participants observed a lot of fruity flavor, banana, pineapple, mango, and only slight phenol flavor. And so it wasn't overpowering. It was actually very, very balanced and very closer to a wheat beer than actually a saison. So that was a very interesting finding, because obviously we don't necessarily think this use can only be used for saison. We think it has so much to offer that it can be used for other type of beer as well. Next slide, please, Jessica. So here you have a couple of other trials from Italy. Again, same taken from the original slide of the 15 trials. So trial 4 and trial 13, and both of them were performed at 26 degrees centigrade, so higher than the previous one. And in this case, and you can see the details for both trials, different type of malt and also different type of hops were used, but in both cases, the profile came back as more of that of a saison beer and less of a wheat beer. Okay, so the peppery note, the spicy notes, the phenol notes were much more apparent at that temperature than at the lower temperature. Next slide, Jessica, please. And in this final trial, was done in Croatia. You can see that there was also some wheat malt used, and that the original gravity was raised by using some glucose in order to reach again a higher attenuation. Then the batch was split into two, and then in parallel they did one fermentation at lower temperature and another fermentation at higher temperature, and they came out with the same conclusion that we've seen in the previous slide, is that at lower temperature you really put the emphasis on the fruit and less on the clove, and at higher temperature you express more the clove flavor and the spicy flavor, so more traditional of a saison beer, and at lower temperature more towards the profile of a Belgian wheat beer. And on this slide side is really the summary of what I just said. So lower depending on the temperature that you decide to use for the fermentation, you can really decide which flavors you want to put forward. Okay, so I think that's very important to know when you decide to use this yeast, that is not just for saison. And on average here, we noted that the attenuation was on average 80%, the pH 4.2, and that, as Jessica mentioned as well, you can reach a final attenuation in 5-6 days. Okay, so these are the results of field trials that were run, with our brewery partners. We have a couple of guest speakers today that will tell us about their experience with this specific yeast, farmhouse, and we'll hear from them in a minute. But maybe Jessica, you can wrap up by talking about what the future looks like in terms of hybrid yeast and what the possibilities are with that technology, again, non-GMO technology.
Mm-hmm. Yeah, this is when I start getting really excited just to talk about what the possibilities are. We're starting with the farmhouse yeast, but basically if it exists within Saccharomyces cerevisiae, so if it exists in wine or beer yeast or even bread yeast, we can breed it. We can't breed things that don't exist in yeast. So we can't breed a bacteria with yeast or a Brettanomyces with a Saccharomyces cerevisiae. But if it exists within the world of brewing or winemaking or cider making, we can potentially hybridize the yeast and create new awesome combinations. So these are just a few of the things that I've been thinking about. So potentially doing a non-H2S lager. So like I talked about, this farmhouse yeast has that naturally occurring mutation that we bred in from that Italian wine yeast. So it can't make any H2S. It can't make any of that rotten egg aroma, which is a major off aroma in lager beers. As I'm sure you guys know, you can come in the morning and smell your tanks fermenting and it smells like rotten eggs. So we can potentially breed this technology into lager yeast. So you can make very clean lagers. Also, maltose negative strains for doing low alcohol beers. I know low alcohol beers are becoming definitely more prevalent, and they're starting to come onto the radar of a lot of craft brewers, with the millennial generation and Gen Zs trying to be more healthy and reduce alcohol consumption. I think you're going to start seeing a lot more low alcohol beers. So a lot of those maltose negative strains currently that are on the market are non-brewing strains. They're wild strains or strains that don't behave like a brewing strain. So creating a maltose negative brewer strain, it'll behave just like a brewing strain, but it can't consume maltose and won't create any alcohol. Potentially doing enhanced ester production, again, for doing dealkalized beer. A lot of dealkalization methods, they strip out the alcohol, but they also strip out all the esters. So we can create a yeast that has, 10 times, 20 times the amount of ester production, so that after dealkalization, your beer still smells nice and estery. Lower VDK yeast for lagers and English ales for quicker tank turnaround time. Creating biotransformation yeast, similar to the wine industry, uses Sauvignon Blanc wine yeast for enhancing flavor and aroma of Sauvignon Blanc wines. We could do that to create an IPA yeast. And then also product-specific developments. So creating something such as seltzer yeast. These seltzer fermentations, I'm sure as you know, are just basically sugar and nutrients. So creating a yeast that thrives in those environments and does really well.